Welcome back, everybody, for part 2 of um, chapter 3, talking about blood banking reagents. So in our last meeting, we ended on this slide of the PowerPoint for chapter 3, and we had finished reviewing differences between the direct antiglobulin test, also known as the DAT, compared to the indirect antiglobulin test known as the IAT. And so just to reiterate where we left off last time, remember that the DAT is used to detect sensitization that has happened in vivo in the individual. And so you will recall from last time that basically here we see red cells that were drawn from the individual, and they are already sensitized with antibodies coating the surface of the red cells. Okay. The indirect antiglobulin test, on the other hand, is something that we use in vitro to help us detect weak reactions or help us detect when sensitization has occurred. So for the indirect test, it requires us to add an antibody source such as the patient's serum or plasma with some red blood cells that we're using for testing. And then we can see here that sensitization has occurred, but since there is no cross-linking between antibody targets to different red cells, we're not able to visualize it. So the antiglobulin reagents, namely here anti-IgG, bridges that gap and targets the IgG molecules that are attached to the red cells in our test system. And now we are able to see the cross-linking and visualize that immune complexes had formed. Okay, so that is where we left off last time. And we're going to move on and we're going to build on the antiglobulin test and talk about something next called check cells. So check cells are also referred to as Coombs cells. And remember, Coombs is the scientist who basically came up with the idea of antiglobulin testing. Check cells or Coombs cells are required by the American Association of Blood Banking as a control system whenever AHG results show a negative reaction. Okay? And so meaning that whether it's a DAT or an IAT, we're not getting agglutination. And so these reactions are negative. So it's basically telling us that sensitization has not occurred in our test system. Okay, so we're only going to use these check cells on negative reactions. Okay, so let's talk first. I'm going to jump down here and talk about what check cells actually are. Check cells, once again, are type O red blood cell reagents that come prepared from the manufacturer with IgG antibodies attached to them. Okay, and so here I drew a very, very basic picture of, we have a red cell, a check cell, and basically it has human IgG attached to it. And that's what a check cell is. So it comes in the vial with the IgG already attached to it. Okay, so when we add these check cells to a negative reaction for the DAT or the IAT, the check cells should agglutinate the anti-human globulin that's freely flowing in that tube. Okay, so think about it. The anti-human globulin, anti-IgG, is not bound to anything on the red cells. Okay, we're seeing a negative reaction. So when we add check cells that have IgG, human IgG attached, this reagent should say, hey, there's my target, and bind to the IgG that is affixed to the check cells. So when we are doing antiglobulin testing, we can get false negative reactions. And we're gonna look at a couple of reasons why we could get false negative reactions in our test system. Okay, so maybe we did not add AHG reagent to the tube. We thought we did, but we didn't. And when you're doing a lot of tube testing at once, it would be very easy to skip a tube. Okay, so if AHG, anti-human globulin, anti-IgG, isn't in the tube, we're going to get a negative result, right? It's going to look negative because the AHG isn't there to bind to anything. Maybe we did add the AHG, but for whatever reason, it is failing to react. Okay, faulty reagent. Or the last reason, which is really very interesting, maybe we failed to wash our tubes properly before we added the AHG. Okay, and so when we review anti-globulin testing in upcoming laboratories, I will review this with you guys again and stress it. Whenever we are going to add anti-globulin reagent to a tube, we have to wash what's in that tube first. And so in the last lecture, I mentioned to you all that washing removes unbound proteins such as IgG that's floating around in the tube, or complement proteins that are present in the tube. Washing removes them so that they're not going to react with our AHG when we add it. Okay, because if we do not wash properly or we do not wash, our AHG reagent can be neutralized by junk that happens to be in that tube. And we're going to build on this in a moment. Okay, but for now, remember, check cells have IgG affixed to them. It's a reagent that we add to any antiglobulin test that is negative. And when we add the check cells to a negative antiglobulin test, we're going to centrifuge again for 15 seconds and we're going to read those tubes. We should see agglutination, and basically it's ensuring that AHG was added. First of all, the AHG is reacting and that it wasn't neutralized. Okay, so here's our check cell review. So basically, we have our anti-human globulin, or anti-IgG, okay, and we added red cells. Okay, we have red cells that we added our anti-IgG2, and there really wasn't any IgG bound to these red cells. They're uncoated. So when we add the anti-human globulin, it had nothing to bind to. So in this test system, it's looking negative. We're not seeing any type of agglutination at all. So this would be a time when we would need to add the check cells. And so here the check cells are a different color. They're hot pink red cells. Okay, so you can see how they have IgG already attached to them. They come that way from the company. So when we add our check cells into this tube, okay, where there's no sensitization that previously occurred, what should happen? Our anti-human globulin, anti-IgG, should then say, hey, there's IgG, that's my target, and bind to it. And so the end result is, we're going to see agglutination after adding the check cells because our AHG is binding to the check cells present in that tube. There was no binding that happened with, the other red cells in the test system. So this is basically added to ensure that AHG was added, the AHG is functioning. And what it tells us, though, is that it's truly a negative test for whatever we were testing, whether it's a DAT or an IAT. So this is a picture that I took of reagents related, of AHG reagents. And so here we see our different anti-human globulin reagents. And let's start with this one on the far right. So here we see this one tells us on the label it's polyspecific. And you can clearly see here it's telling us that this is antibody against IgG and C3d. Okay, so it's polyspecific. It has more than one antibody present in it. Okay, and so typically when we're doing anti-human globulin testing, we start with the polyspecific. If we get a positive with polyspecific, then we need to go on and test with monospecific to find out whether IgG or complement are coating the cells. Okay, so here we see two different bottles of monoclonal anti-IgG. I didn't have a bottle of anti-C3, so you're not seeing that one here. And what you're going to notice is the second bottle is a pretty color. It's kind of like an aqua green sort of color. Some labs really do prefer to use a bottle of AHG that has a pigment to it. And similar to reasons why anti-A and anti-B have colors, okay, it's basically we know we added it when we see the green color in the tube, okay, so it reduces an error of missing adding it to the tube. On the very far left, we see our check cells here, and I love the company that we buy them for has a little check mark there, and if you read the label, it is telling you exactly what I told you check cells are. So they are IgG coated pooled red cells. And what ABO group do you think they are? I wish I could ask you guys this in person. Hopefully you would tell me they are type O. They must be O because we don't want A or B antigens to get in the way of our test system. So they have the IgG attached to them already. When are we going to use check cells? When we have a tube that had antiglobulin reagent added to it, whether it's polyspecific or monospecific, that is testing as negative, okay? We add a drop of the check cells to the negative tube, centrifuge and read. We should then see agglutination, and we grade the reactions of our positive check cells as well. They're going to be weaker, 1+ maybe reaction. But what this tells us is that the tube test is actually truly a negative test. The check cells are just confirming that the reagent was added and that the reagent is functioning properly. So let's take a look at some sources of error in antiglobulin testing. And this first table, 3.9 from our textbook, we're going to review false positive reactions in antiglobulin testing. Okay, so we're getting positive results, but they shouldn't be positive. Okay, so false positives. So first one here, the red cells in our test system were agglutinated before the washing step and the addition of anti-human globulin reagent. Okay, so that's clearly going to cause a false positive, and it could be due to antibodies that a patient has. The use of dirty glassware, if there's particles or things present in the glassware, it could look like a false positive. Improper centrifugation of the tubes, and here if we over centrifuge, okay, so basically over centrifugation means centrifuging for a longer amount of time than necessary, or maybe at a faster speed than necessary, could lead to a packed red cell button that looks like it's positive when we're trying to read the tube. And so just to reiterate, when we're centrifuging to read reactions in tube testing, we only centrifuge for 15 seconds, and the centrifuge speed is 3,500 RPMs. And in our lab, in our student lab, we have specific centrifuges designed for blood bank testing that are already preset to 3,500 RPMs. Okay, so if we centrifuge too long, it could look like a weak false positive. So next we're going to look at table 310, which is going to outline false negative errors that can happen in antiglobulin testing. And the first one is really very important. I'm going to just mention it briefly now, but we're going to spend some time on another slide in a minute looking in more detail how this actually happens. So mentioned to you all a few minutes ago that before we add any type of anti-human globulin reagent to a test tube, whatever's in that test tube needs to be washed to remove unbound proteins that could be present in the tube. If we do not wash adequately, okay, unbound protein junk that's present in the tube could bind to our AHG reagent, and this is called neutralization. And what happens is it looks like a false negative in our test system because these junk antibodies aren't attached to a red cell. So if the AHG just binds to some antibody that's floating around, we're not going to see agglutination in the tube. But basically these binding sites are taken up, so they're not able to bind to sensitized red cells that might be present. And we're going to look at a slide about that in a minute. So improper washing leads to false negatives. We could get false negative if our testing is interrupted or delayed, if the AHG isn't added immediately after washing. Okay. It could look like a false negative if we're not identifying weak reactions, and this would be a technical error in our reading. Our AHG lost its reactivity, okay, due to improper storage or contamination. As mentioned before, maybe we did not add AHG reagent. That's another technical error related to our testing. Once again, centrifugation can play a role here, and here it's under centrifugation, which means centrifuging for a shorter amount of time, maybe at a slower speed, is leading to the false negative. So maybe we're not centrifuging for 15 seconds less, we're going to get a false negative result. And then lastly, if we're using red cells in a concentration that is not in our optimal range of 2 to 5%, we could be missing agglutination reactions. So weaker red cell suspensions could also contribute to false negatives in this testing. So this is a very crude, basic slide because I am not a graphic designer and I made it. So you can have a little laugh at it, but hopefully it's going to clear up any confusion you may have about neutralization. Okay. So let's look at this picture. So in our test tube, we have some red blood cell and it has IgG attached to it. Okay. So these are sensitized red cells. Now, we talked about how it's very important to wash properly before we add our AHG. And in student lab, we always wash 3 times. Okay, and what we're washing with, maybe I didn't mention this to you all before, is physiologic saline, because that is something that is not going to adversely affect the cell membrane of the red cells and cause hemolysis. And so you guys will get to do some of this testing actually, even though this course is completely virtual and online. When we come back, hopefully in the fall, you know, we'll have 1 or 2 labs and blood banks just so you guys get some hands-on with doing some of the techniques. So anyway, if we don't wash properly, look what's hanging out in the tube as well. Okay, this is some IgG molecule, but you notice it's not affixed, it's not coating any red cells. So this is basically a junk antibody. Okay, so if we don't wash properly with saline and have this get thrown out with the wash, it's going to remain in the tube. So that when we add our anti-IgG, and you can see I have it a different color, it's a brown color here. When we add our AHG to the tube, AHG isn't smart enough to know which IgG molecule to bind to. It basically just binds to the first one it comes into contact with, because both of these are human IgG. It's just one isn't attached to a red cell and one is coating a red cell. Okay, so failure to wash properly, the binding sites of our anti-IgG could be taken up by junk antibody, and basically this is going to look like a negative when we centrifuge this tube, okay, because this is not attached to a red cell, so we're not going to see any agglutination here. It's going to look like a negative result. So this is called neutralization because the 2 binding sites of our anti-IgG are taken up by the junk IgG antibody, so they're not available to bind to their target IgG that's actually coating a red cell. So in essence, sensitization has occurred in this tube, but we are not detecting it because our reagent was neutralized. So it looks like a false negative in our tube. So let's look at one more slide to do our review. Okay, so check cells are, remember, added to negative AHG reactions. We do not add them to positives. They are only added to negatives. They should agglutinate, okay, when they are added to a negative tube. They are going to check that the tube was washed sufficiently, that AHG was indeed added, that the AHG reagent had reactivity. However, it does not ensure that the AHG reaction is negative, okay? So if washed properly, AHG was added and AHG is functional, agglutination will occur in the previously negative tube, okay? So here we have our check cells, okay, and they have IgG attached to them already. And here we have our anti-IgG that's going to bind to the check cells. So the check cells are going to be agglutinated. Okay. So we're going to move on to a different topic now. And these are reagents called potentiators that are used in blood banking. And this is table 3.12 from our textbook, which lists a bunch of different potentiators that can be used in blood bank and basically how they affect reactions. But before we review this table, I want to point out that potentiators are simply reagents that enhance the detection of IgG antibodies by increasing their reactivity. Okay, so as we go through, hopefully this will make a little bit more sense. So the first potentiator listed is called low ionic strength saline, or sometimes people call it low ionic strength solution, and it's abbreviated as LynnC. So people refer to it as LynnC. LynnC basically enhances reactions by increasing the rate of antibody uptake. Okay, so when we have a test system, not used for ABO, okay, maybe during an antibody screen or an antibody panel, we want to enhance these reactions that tend to be weaker. We can add 2 drops of LynnC. So anyway, getting back to LynnC. So basically, LynnC increases the rate of antibody uptake, that first stage of agglutination sensitization. So we add 2 drops of LynnC to our tubes, and it's going to help these antibodies that are maybe a little bit weaker bind more readily to their target antigens. Okay, and we use LynnC in our student lab. The second one is called polyethylene glycol, or PEG. PEG concentrates the antibody in the test environment in LynnC. So PEG is not usually used unless we know that we have a really very weak reaction. The next one, interestingly, proteolytic enzymes, and the 2 that are commonly used in blood bank are papain. This one happens to come from the papaya fruit, if you eat papaya, and physin. So you can look at the description here and get an idea about how these enzymes work. So they basically break down proteins, and they remove negative charges from red cell membrane, reducing the zeta potential. However, using these enzyme-treated cells, some red cell antigens are destroyed by the use of the enzyme treatment. And we're going to be talking a lot more about this one a little bit later in Module 2. The last one here, bovine serum albumin, reduces the repulsion between cells but does not shorten the incubation time. So basically it shortens that zeta potential, and so binding can occur more readily. So what do you need to know about potentiators? Know that they are just reagents that we use to enhance detection of IgG antibodies. There are quite a few of them that can be used in our test system. Okay, I'm going to move on to another group of reagents called lectins. Lectins are actually very interesting because they react in the same way as antibodies. However, they are derived from plant sources. Okay, so these are seed extracts that have a specificity toward and react with certain red cell antigens similarly to how an antibody would react, but they're not antibodies, so they're called lectins because they come from plants. They bind to carbohydrate determinants of red cell antigens just like an antibody would. Okay, and there are a few that are very important to blood bank testing. So this first one, an antibody against A1, is the most common one that's used, and it is derived from a plant. If anybody took Latin, I doubt it. I took it a long time ago, but it's called Dolichos biflorus, and this is actually the sweet pea plant if you're curious and you're interested in plants. So this extract from the seeds of this plant, it's used in the same way an anti-serum would be used. Anti-A1 is going to bind the A1 antigen that most people who are type A have. And when we get into ABO groups, we're going to learn more later that you can be type A. The most common type of A is type A1, but there's a significant portion of the population that is A2. Okay, and so anti-A1 from Dolichos biflorus is only going to agglutinate those people that are A1 antigen. Eulex europaeus is antibody against a big H antigen, which we'll talk about a little bit later. Vicia, I can't say this one properly, graminia is antibody against big N antigen, and Iberosamara is antibody against big N. So we'll talk a little bit more about these later. Just remember that lectins come from a plant, but they work in the same way in binding certain antigens as an anti-sera would. So just a couple more things to do with this unit. We're going to basically review other methods in blood banking that are available for testing. And what I can tell you is that when students are learning blood bank, you always learn the tube test first. The tube test is basically the classic way of doing testing. However, when you're working in a busy blood bank laboratory, it would be just really maniacal to think that you could test every donor or every patient with tube alone. You'd be sitting there all day. You'd go crazy. Okay, so there are automated methods that have been developed to test larger volumes of patients, and one of these newer technologies is called the gel technology. We actually have this in our student lab at CCP. So when we get together for 1 or 2 labs just to do a little bit of testing, so you guys get some hands-on, hopefully in the fall, you'll get a chance to do some of this gel testing. So basically, this is a picture of a gel card, and the card has 6 microtubes in it. This is about the size of a credit card. Okay, so it's enlarged here, and each microtube has these polyacrylamide or dextran acrylamide gel particles in the bottom of the tube. And so basically, we add a certain volume of red cells or plasma or serum to each tube depending on what we're testing for, and then we centrifuge the card. So you need to have a special centrifuge that can accommodate the cards, and we have one at CCP. You guys will get a chance to see it and use it at least one time. And the bad thing about the centrifugation, what I can tell you is when you're centrifuging tubes in tube testing, you heard me talk today about how it's only 15 seconds and we read it and we look for the chunks of agglutination in the bottom of the tube using a viewing lamp and a mirror. 15 seconds is fast. These gel cards centrifuge for 15 minutes. 15 minutes, okay, long time, okay? But here's what we're going to find when we're done, okay? So when we have the strongest agglutination reaction possible, a 4+. The red cells, have agglutinated, and think about it, they're forming a big mass of particles, and so they're too big to sink through the gel. So a 4+ reaction remains completely on top of the gel. It's too big to sink down. 3+, if you look here, we can see we still have quite a lot of agglutination on top, but we're starting to see a couple of pieces sink through the gel. That would be 3+. 2+, we could see that the particles are interspersed throughout. 1+, we could see that there's a good portion of particles here on the bottom, but you can see how there are some trapped in the lower portion of the tube. And then a true negative is going to be a button in the bottom of the tube. So you have to be very careful when you're reading gel, because when you're reading tube, a big button in the bottom of a tube is usually a 4+ reaction. Okay, so you have to keep it straight. 4+ in gel is they're trapped on top. Okay, so here this would be a negative reaction designated by a 0. Now there's one other thing at the end of the card that I'd like to just talk about for a minute, and we'll be getting into this more as we go through our units. When you see MF, MF describes a reaction known as mixed field. Mixed field reaction means that when we're looking at either a gel tube or even the test tube method, we're seeing some cells that look like they are agglutinated, as in a positive result, but then there are other cells that are present in the test system that look negative. Okay, so let's look at this mixed field tube here. So if you look on top, you can see that there's a thin layer of cells on top here, okay, that look like they would be 4+, but then there's also some cells on the bottom that look like negative. So two very divergent results within the same tube. We call this mixed field. When would mixed field occur? Mixed field can occur, for instance, if a patient has been recently transfused with a unit of donor red cells, and so maybe the donor red cells are agglutinated and their own cells are not. Okay, but also a perfect example of a mixed field reaction would be when we're using when we're doing an anti-human globulin test that tests as negative and we're adding check cells. Okay, so let's think about this. This is a good way to review our check cells. So with our anti-human globulin test, the red cells that were originally in the test system did not agglutinate, okay, and it looks negative, so those cells would be on the bottom. And so we just learned that with AHG, whenever we have a negative, we have to add the check cells, and the check cells are coded with human IgG. So when we add the check cells into the system, they're going to agglutinate with the AHG, and so they might be on top. Okay, so usually mixed field means two populations of cells are present, and one population of the cells is reacting, and the other population of cells is not. Okay, so that's mixed field. So this is a drawing basically outlining what we just described on the last picture. So 4+, completely on top, 3+ mostly on top, 2+ throughout, 1+ towards the bottom, and negative complete button on the bottom. Okay, so this is a picture, and it might be a little bit hard to see. It's kind of a blurry picture that I found on the internet. It's interesting. It's showing you ABO grouping, okay, in a gel card. And so what's really interesting about the gel cards is, can you see how this tube is blue? The gel card already comes with the anti-A added to that well. The B has the anti-B, which is yellow, added to it. The D has the anti-D already added to the tube, so there's no need to add any anti-sera there. And so basically, we have our forward grouping. Okay, remember forward grouping, we're using known anti-sera here, anti-A, anti-B, and anti-D, to find out which antigens, if any, the patient has on their red cells. Okay. The control here is a well where we just add the patient's red cells to the patient's plasma. And this patient control is really done to make sure that the patient is not auto-agglutinating. They're not agglutinating, their own cells. Can also detect if some type of sensitization has happened in vivo in the patient, similar to a DAT, okay? And then we get into our reverse typing. So reverse typing, to review a little bit, is when we're using reagent red cells, A1 cells in this well and B cells in this well, to determine which naturally occurring ABO antibodies the patient has, okay? And so remember Landsteiner's rule. We all have naturally occurring ABO antibodies to the antigens that are not present on our own red cells, okay? So let's take a look. So we look at this first tube, anti-A with the patient's red cells, and we see that they're completely on top here. This would be a 4+ reaction, okay? So what does this mean? Our patient has A antigen, okay, because it agglutinated with the anti-A. When we look at the anti-B well here, we see that the patient's cells are all the way here on the bottom, and in gel testing that means negative. So this means no B antigen. When we look here with the anti-D, we could see once again a 4+ reaction of the patient's cells on top. So this means that the patient has the D antigen that reacted with the anti-D in the tube. And so basically, what I like to do in the beginning is just look at the forward typing and say, okay, well, what type does my patient look like based only on the forward typing? And this, we're going to say A positive, okay? A and D. So remember, D is the one that tells us about the RH. The patient control is negative, which is good, which means we're not having any kind of weird agglutination, patient agglutinating themselves, which could, mask the true reactivity of our test system. Then we get into the reverse typing. So with reverse typing, remember, we want to find out which antibody, naturally occurring ABO antibodies the patient has. So I always try to reverse my thinking and think it through. So if this patient has A antigen, they should not have anti-A, they should have anti-B, okay, because they lack the B antigen. So let's look what we have. So here we see no reaction with the A1 cells, meaning that the patient does not have anti-A in their serum or plasma, okay? And when we look at the B cell tube, we can see a 4+ reaction here, indicating that this patient's plasma has anti-B that agglutinated these B cells, and 4+ reaction is seen on top. So when we're doing ABO typing, we always want to make sure that the forward and the reverse agree, because if they don't, there's a mismatch. It's a discrepancy, and we have to figure out what's going on. Okay, we're going to be building on that when we get into ABO testing in future module, not future module, future lecture, okay? So here, this gel card is telling us the patient is A positive, they have anti-B, and that makes sense according to Landsteiner's rule. So we're going to move away from gel method, and we're going to talk a little bit about testing done in microtiter plates. And when you're working at a very large institution, and this is a shout out for Christine, so the Red Cross basically processes hundreds and hundreds of donors on a daily basis, and they're not going to be sitting there doing this in a test tube. Okay, so they use microtiter plate technology of some sort. And here we see 96 well microtiter plate, and it's very automated at the Red Cross. So, red cells and anti-serum are all added, with a robotic arm into the plate. The principle here is the same as the tube method, okay? So testing is just going to take place in a smaller scale instead of in a tube in the well of a microtiter plate. So with this method, a concentrated button indicates a positive reaction, okay? So what do we mean by a concentrated button? It looks like this in the well. And actually, at the Red Cross and other blood donor centers or high volume labs that do this type of testing, they actually take pictures of it and it's interpreted, you know, through photographic imagery. Okay. So this is what a positive would look like in the well. A negative would be dispersed cells. Okay, so they're not gathered in the center. Okay, so our next method is a little bit different. It's called solid phase. It also uses microplate wells, but it also uses immobilized reagents. And here, the interpretation is a little bit different. It's the opposite, okay, of the microtiter plate method we just looked at. So here, with this solid phase, cells that adhere to the sides and bottom of the wells are positive. Okay, so here you could see that dispersed cells are positive. Cells that form a button would be a negative. So you could see how interpretation varies depending on the method that you are using. So we could look at this whole strip here. This would be negative, 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 positive, 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 positive. Okay, and once again, the reactions can be graded. So the strongest reaction is when everything is really dispersed in the well, 4+. 3+, you can see 2+, 1+, and then negative. Okay. This is also done on instrumentation, and pictures are taken for interpretation. Okay, so other methods, number 6 for our solid phase. So what do you guys think the patient type is? I really wish that I could be with you to hear your answers. Okay, so with solid phase, remember, when we see this dispersion of the red cells throughout the well, that's a positive. So we have a 4+ here with the anti-A, a negative with the anti-B, a 4+ here with the anti-D. Okay, so based on the forward typing of solid phase here, this looks like A positive. So remember Landsteiner's rule. Someone who is type A should have a naturally occurring antibody against B antigen. And here we see, yay, a 4+ reaction with B cells, indicating that they have anti-B in their plasma that reacted with our reagent B cells. The A1 cells are negative, which makes sense because if they are indeed type A, they should not have an antibody against A agglutinating their own cells. So sorry, I really searched far and wide to try to find a different type for this method, but I couldn't find one. So you get the same one, A positive on gel and A positive on solid phase. So that basically finishes up our discussion of part 2. I just wanted to mention that throughout the course, there are some other free resources available to you guys. And I've had students really check out this website quite a bit in the past, and everybody raves about him, and I do too. He calls himself the blood bank guy. And he's basically, I think he's an MD, PhD. He's a blood bank specialist. And so I think like in the late 90s, he started realizing that blood bank could be a pretty confusing topic for a lot of people. And he put together different free resources on his website, which we'll look at real quickly in a minute. But he has videos on different topics, practice quizzes, podcasts. And so you see the link here. And I also included the link on our homepage. If you ever want to hear someone else, you know, describe or outline some of the topics we're talking about, please check out his website. I'm going to end this slideshow, and I'm going to stop the share, and I'm going to go real briefly to the Blood Bank Guy's website, and Okay, so here's the website of Blood Bank. Oh, here he is. Looks like a nice guy. Okay, so when you go to his website, you can see up here, he talks about himself. There's a blog, there's a glossary of terms. He has different podcasts, quizzes, and videos. I'm just going to go to the videos real quickly. And so these are topics that are really considered essential. Some of them we haven't really gotten into next, but you know, blood groups, okay, pre-transfusion testing, antibody identification. These are definitely things that we're going to get into a little bit later, but he definitely has quite a few different ones out there. Sorry, I don't want to make you dizzy, that you can check out. And I'm not going to really play these for you guys because I know when I, you can see some of them are a little bit old, but he basically lists all of them here. And then there are quizzes. So I know you guys love doing practice questions, and I think I mentioned to you guys previously that our blood bank textbook, each chapter really does have excellent multiple choice questions with the answers in the back that you should be doing, you know, to make sure that you understand the topics. And then he has quizzes. So this one might be a fun one. So, you know, in our upcoming unit, I think in lecture 5, we're going to start getting into ABOs, more about them, and this is a quiz that you can take to make sure you understand everything about ABO. He has one for RH when we get into those in chapter 6, and he has other blood groups that we'll be discussing as well. So, you know, it's kind of nice that he has different quizzes out here to help. Okay, so check out his website. I'm going to stop my share there for today, and I'm going to stop our recording. And, you know, if you have any questions about any of this material, please remember that we have a discussion board for each module. Post your questions on the discussion board, and if any of your peers, if anybody can answer the question, guess what? You might earn 5 points. But I'm also available to answer questions too. And if you're really having difficulty, you know, please message me. We could even set up a Zoom meeting, you know, if you're really having trouble. Okay? So good luck, and I'm going to stop our recording.